KCNQ1 and Long Q-T syndrome. Daniel Bergart, Long Q-T syndrome. LQTS is a disorder of cardiac repolarization resulting in QT prolongation on an electrocardiogram. 11. 12. But LQTS can lead to many other symptoms, some far more serious than a longer QT interval. Sudden loss of consciousness. Seizures. Sudden death. What causes these phenotypes? Many reasons, some unknown. The main reason is mutations in the KCNQ1 protein. Chromosome placement 16. KCNQ1 a potassium voltage gated channel, KQT-like subfamily, member 1. Helps shuffle potassium ions across membrane to repolarize heart cells. Must be phosphorylated in order to function. KCNQ1 protein structure 15, phosphorylation sites and KCNQ1. There are at least 7 activating phosphorylation sites along the KCNQ1 protein 15. Ion_trans. KCNQ_channel. 100. 200. 300. 400. 500. 600. S27. S409. T513. S577. T470. S464. S468. 676R. What phosphorylates KCNQ1? PKA2A/B, PKA2A, PKA2B. But they need help attaching to target proteins. A Cap9 protein. 14. A Cap9 is a scaffold protein. The needed protein to attach the PKA2A/B complex to KCNQ1. Previous experiments. And the one I chose to challenge. Why use mouse as a model organism? PKA2A. A Cap9. PKA2B. KCNQ1. Experiment main hypothesis. The rebinding site in a Cap9 is important in the phosphorylation of KCNQ1. Experiment Part 1. Establish that a Cap9, PKA2A/B, and KCNQ1 localize to the same areas. Experiment Part 2. Test to see if changes in the Cap9 rebinding domain affect protein binding. Experiment Part 3. Screen for unknown mutations for further testing. Do all 4 proteins localize to the same part of the heart cell? Method: immunofluorescence analysis of heart cells. Protein. Antibody with fluorophore. Localization of KCNQ1 in heart cells 5. Are all 4 proteins active in the same cell functions? Method: look at gene ontology terms. PKA2A3. A Cap92. PKA2B4. KCNQ11. Experiment main hypothesis. A mutation in the rebinding site in a Cap9 causes a decrease in phosphorylated KCNQ1. Experiment Part 1. Establish that a Cap9, PKA2A/B, and KCNQ1 localize to the same areas. Experiment Part 2. Test to see if changes in the Cap9 rebinding domain affect protein binding. Experiment Part 3. Screen for unknown mutations for further testing. Does changes in a Cap9 rebinding sites affect the binding of PKA2A/B? Method: microarray of mouse strand a Cap9 protein to look for phosphorylated PKA2A/B complex. A. Weight a Cap9. B. A Cap9 delta rebinding site. Rebinding domain. Shows way to Cap9. Shows a Cap9 delta rebinding site. Experiment main hypothesis. A mutation in the rebinding site in a Cap9 causes a decrease in phosphorylated KCNQ1. Experiment Part 1. Establish that a Cap9, Ca2a/b, and KCNQ1 localize to the same areas. Experiment Part 2. Test to see if changes in the Cap9 rebinding domain affect protein binding. Experiment Part 3. Screen for unknown mutations for further testing. Is there a specific mutation in the rebinding site on a Cap9? Method: DNA sequencing of patients with congenital long Q-T syndrome, LQTS. Future directions. Is the mutation that is found in the rebinding site causing a problem in localization of the Ca2a/b complex? Does the same interactions of proteins occur in all cells known to have these proteins? Is there a mutation in the Ca2a/b complex? Questions.